Hello. I can take off the mask. Hello, everybody. Good evening. Good evening and welcome to tonight's Friday evening lecture. My name is Rudolf Oldenburg, and I'm a senior scientist here at the Marine Biological Laboratory. You might notice that I'm reading from prepared text, so I don't stray off too much. Thank you to those who have come out to the Cornelia Clapp Auditorium and those who are joining us virtually. Throughout the summer, we are recognizing past members of the MBL community who have made extraordinary contributions to this storied institution. Tonight, it is my privilege to share with you highlights of the life and work of Jean Clark Dan, a student, faculty member, investigator, and corporation member of the MBL, along with her husband, Katsuma Dan, also a faculty member, investigator, and corporation member. Both Jean and Katsuma first came to the MBL in 1931. Katsuma was a graduate student of Victor Heilbronn in developmental biology at the University of Pennsylvania, and it was his use of sea urchins and other marine animals that brought him to the MBL. Jean was a biology student at Wilson College in Pennsylvania and came to the MBL for an invertebrate zoology course before pursuing graduate work, also under Heilbronn, at the University of Pennsylvania in 1932. Despite Katsuma's and Jean's very different backgrounds, one, the son of a noted Japanese business and statesman, the other, a middle-class American from New Jersey, their shared interests brought them together, and they soon became both scientific colleagues and romantic partners, becoming engaged in 1934. While Katsuma returned to Japan to continue his research at the Misaki Marine Biological Station, Jean remained in the United States to finish her PhD. A month after she completed her thesis in 1936, Jean and Katsuma were married here in Woods Hole. Soon after, they settled in Japan and continued their research together at the Misaki Station. Despite the darkening clouds of political upheaval and impending global conflict, Katsuma and Jean managed to find refuge in each other and in their work. That work was challenging enough in its own right. Much about the fundamental processes of cell division and differentiation was still cloaked in mystery. But teasing out those secrets using light microscopy and marine organisms looked very promising. Using polarized light microscopy and working with his graduate student and later MBL distinguished scientist Shinya Inoue, Katsuma was the first to isolate and identify the mitotic spindle that appears during cell division. Jean worked on microscopic studies of the fertilization process, defining the sequence of events that occur when sperm meets egg. Jean and Katsuma also co-authored articles on the behavior of the cell surface during cleavage. Misaki Station was situated at the entrance to Tokyo Bay, about 40 miles from the capital city. Although not a military target, work there was increasingly affected by the war, first by shortages of vital supplies, followed by US bombers within range of Japan. Then, in early 1945, the Japanese military took over the station and transformed it into a base. Katsuma, Jean, and their colleagues were forced to relocate to temporary facilities nearby to continue their work as best they could. When the war ended in August 1995, Katsuma rushed back to Misaki Station to ensure the US Navy wouldn't destroy it. In a note he left behind, he explained that the Marine Biological Station was for peaceful research and compared it to other famous facilities in the US, including the MBL. The American military heeded Katsuma's plea, and Misaki was spared. Katsuma's note, signed "The Last One to Go," now hangs in the library lobby here at the MBL. Jean visited the United States and the MBL in 1947, after 10-year absence. Through her network of friends and colleagues, she acquired the first phase contrast microscope in the US, which she brought back to Japan as a gift for her husband. Katsuma, however, encouraged Jean to use it herself and return to her scientific work. She went on to do pioneering work in invertebrate biology using light and electron microscopy, and discovered the acrosomal reaction, a critical step in the fusion of sperm and egg during fertilization. Apart from their important scientific discoveries, Jean and Katsuma made significant contributions to education. Katsuma was a professor of zoology and later president of Tokyo Metropolitan University, while Jean was a professor of biology at Okanomichu Women's College. Jean also helped found the Teteyama Marine Research Center and taught there with her husband. They both maintained strong connections with the MBL and Woods Hole, and after Jean died, her ashes were scattered off near Nobska Point. We are pleased to honor Katsuma Dan and Jean Clark Dan, who provided a shining example not only of the global influence of the Marine Biological Laboratory, but also the capacity of science to transcend national borders and political hostility for the benefit of humanity. And now, I'd like to turn it over to Dan. He is Purdue Fletcher, Hernando Chatterjee Chair in Engineering Biological Systems at the University of California, Berkeley, and co-director of the MBL Physiology course, who will introduce tonight's speaker.
Thank you, Rudolph, for that wonderful history. It's my great pleasure to introduce our lecturer for tonight, Cliff Brangwynne. Let me start by getting the facts about Cliff out of the way. He grew up in Massachusetts. He's currently a professor of chemical and biological engineering at Princeton University, where he holds the June K. Wu Professorship. He's also an investigator with the Howard Hughes Medical Institute and serves as director of the Princeton Bioengineering Initiative, a new campus-wide multidisciplinary effort to drive research at the intersection of engineering and the life sciences. Cliff is the recipient of numerous awards, including the Searle Scholar Award, Sloan Research Fellowship, MacArthur Fellowship, Wiley Prize, and Blavatnik Award. Now, any one of these awards would point to an outstanding and up-and-coming, an accomplished researcher. But together, they signify someone who is fundamentally changing the way we think about biological systems. How has he done this at such a young age? Don't let the full beard, the erudite elocution, and the grandfatherly kindness fool you. He's young. The answer to his success so early in his career, I think, is that he came to biology as a bit of an outsider with a different mental framework for how the world works. His undergraduate degree is in material science, and his PhD is in applied physics. Those are worlds comfortably based on physical laws where detailed understanding of states of matter have given us materials that are well understood. These are materials that we can use to build cars and airplanes and computers that function as expected. But Cliff chose to delve into the less predictable workings of biological systems and the materials that comprise them. First, through experiments on physical properties of cytoskeletal filaments while a PhD student, and then taking a brave leap to organisms, the model system C. elegans, for his postdoctoral work with Tony Hyman at the Max Planck Institute in Dresden. The materials that make up a worm are far from anything we'd put into a car or a plane or a computer, but surely the laws of physics still apply, right? Well, it's at this question and at this point that the MBL comes in. After being part of the physiology course as a student in 2006, Cliff came back to Woods Hole with Tony for a research rotation in physiology in 2008. And it was here, together with students and collaborators in the course, that Cliff had a key insight while working on an experimental system he had been developing as a postdoctoral fellow. That insight set Cliff and Tony and researchers across the spectrum of biological sciences on an adventure to rethink the mechanisms that drive spatial organization. Now, some might say it was the diverse scientific backgrounds of students and faculty in the physiology course that helped create that moment. Others might point to the social and intellectual fuel of the Captain Kidd and the pie in the sky. Maybe it was simply getting to look at an old experiment in a new way, free from the normal constraints of academic life, and in the presence of open minds from around the globe, something that keeps many scientists coming back to MBL year after year. I suspect Cliff's brilliance and willingness to rigorously investigate new ideas played no small part. Whatever the source, a revolution in thinking about organization has begun. Now, what is this new insight? Well, that's Cliff's to share, if I leave him some time for that. But I will caution you that his talk might leave you walking out of this lecture feeling a bit more like a gelatinous glob than you walked in. But that's okay. Certainly over my month here, with all the trips to pie in the sky, I certainly feel like that anyway. And perhaps the truth, knowing the truth about how we are built, what drives organization, what controls it, and with further insights from time spent at MBL, Cliff and many colleagues will discover new ways to re-engineer biological systems, identify causes of disease, and devise new therapeutic interventions. But the big agenda, no pressure, Cliff. So please join me in welcoming Cliff Brangwynne for the evening lecture.
Thanks, Dan. Appreciate it. All right, can everyone hear me and see this? So thanks so much, Dan, for that nice introduction. I'm actually going to try to stand over here. This is a very special moment for me. It's a great honor to give a Friday evening lecture. It's really something that, it's probably the most excitement that I've had about giving a talk in my career. It's also a special moment for me because this is the first time I've given a talk sort of live in a year and a half or so. So in some ways, this is, I'm worried that I'm going to be very rusty. I've been doing it, behind a computer screen for so long now. But it's really a tremendous pleasure to be here and tell you about some of the things that I'm interested in and some of the history of that work here at the MBO. So what we're going to talk about today is biological organization. And I've got this picture. This is going to be a movie I'll show you in a moment on this title slide. This is a C. elegans worm. It's a very humble organism. You can barely see it by eye. And yet the technology, I'll call it, within this organism is vastly more complex and advanced than any man-made technology that we have. We're all very proud of human technologies and civil engineering projects and computers and so forth, but the technology that's embedded in this simple organism is vastly more advanced than anything that human engineers can do. The technology is vastly more interesting and exciting, and we don't understand it. I'm going to show you a movie now, which is the embryo developing into this worm. It's sped up many times. It's a time-lapse movie. This is a one-cell C. elegans embryo that undergoes successive divisions, 2 cells, 4 cells, 8 cells, and so on. Now we're at a many-cell stage. The cells within the embryo are elongating and you start to see something maybe that's beginning to look like a worm, this worm will eventually break out of the eggshell. It'll hatch, it'll crawl around, it'll feed, it'll reproduce, it'll sense its environment. Now, what's, I think, just sort of mind-blowingly exciting about this is that this was really a bag of molecules that somehow spontaneously self-assembles into life. It self-assembles into what is basically the most complex form of matter in the known universe. That's something that we don't understand yet, and it's something that I think is part of a great challenge of biology. Now, we'll see if this advances. So I'm talking about living systems as a kind of technology, and when I say that, this is a picture that might come to mind. We're thinking about an individual cell as a form of technology, a kind of machine. My laser pointer doesn't work, but I was told that the virtual people wouldn't see it anyway, so I'll just describe what I'm pointing to. So this is the kind of picture that might come to mind if I speak about biological systems or cells as technology. You might picture a kind of a machine like this with gears and something like a watch or a car that has these interconnected pieces that are moving in very specific ways. That's an image that comes down to us and has been really prevalent in our thinking about biological systems for many years. And thinking about technology, that's the kind of image that comes to mind. There's a long history of thinking about living systems from those kinds of analogies, dating back to the Industrial Revolution and the kinds of thinking that early pioneers in biology had. This is a so-called lock and key model for biomolecular interactions that comes to us from Emil Fischer in 1894. And this has been an incredibly influential picture. The idea is that biomolecules fit together in very specific ways through the complementarity of their shapes and charge and so forth, just like a lock and key, or if you like Legos, if you're putting Legos together. He was thinking about it in the context of enzyme substrate interactions, but it's really been a dominant picture for the last century in thinking about how molecules come together to build living systems. So that picture has been incredibly fruitful, and in fact, much of our understanding of the building blocks of life is built around that idea, the shape giving function or structure function. These are some examples of the biomolecules within all of our cells and the way their shape allows them to fit together to do very specific things. I could give you many examples of this in biology. There are really too many to count, but probably most appropriate for the first talk I'm giving in a year and a half after this global pandemic that we're still fighting, we can think about the SARS-CoV-2 virus, the virus that causes this COVID-19 pandemic. We're all familiar with this artist's rendition of the structure, and we have these spike proteins that it's a trimeric protein assembly that fits together with receptors on human cells in very specific ways to enable the virus particle to bind to a human cell and ultimately get into the human cell and reproduce, make more viruses and spread. So this idea of parts fitting together in very specific ways is really dominant in our idea about how living systems are built and the way in which they function as kind of technologies like a car or a watch or things like that. So one starts to have an image like this. Okay, we think about humans, we think about organisms built from parts that fit together in very specific ways. There's a problem with this picture, though, in that it's clearly a pretty static picture. In reality, there is incredible dynamics at the heart of molecular interactions. There are also not just one or two or three or a dozen, but tens of thousands of different types of molecules all residing within the same environment within a living cell at the same time. All those molecules are dynamically fluctuating and interacting with one another. This is a simulation that I'll show you, which shows different molecules, proteins and RNA, all jiggling around and interacting. This is a beautiful simulation. It's also incredibly misleading, and I'll try to convey to you why that is. This movie is showing you only about a microsecond of time. So it's a very small snapshot of time. What that means is, in reality, each one of these proteins and RNA molecules is actually rotating around in changing axes about 1 million times a second. Each one of the molecules is interacting with adjacent molecules at a rate of about, I'll say, 500,000 per second. If you calculate the velocity at which, instantaneous velocity at which these molecules are moving, you'll get numbers of about 20 meters per second. So that says that the molecules would zip across this room in a few seconds. And yet they're all on this tiny scale. So the dynamics are really sort of mind-blowing if you think about what's happening in all of your cells right now. Tens of thousands of molecules rotating around millions of times a second. So the picture of their interactions, their sort of interactions fitting together in very specific ways is like the machines we're used to, cars and watches, is very misleading. The analogy that I would say one could make is, we have a pretty good list of the parts in cells. We've got very detailed proteomics, for example, list of all the proteins in cells. It's like saying, well, we have a list of the parts that make up a car. That's very different than understanding how a car works. And of course, a car is vastly less complex than a single cell. The only analogy I think I can make is that if we took all the parts that made up a little car and we put them in a box and we violently shook them, maybe like in an earthquake, and then somehow a car drove out of the box. That's basically what's happening, like in the movie that I showed you in the beginning, where this bag of molecules and somehow a worm pops out. That's what we're talking about when we as scientists look on the processes that make up life with awe, because it's that amazing. It's that far from what we can understand based on the kinds of machines that humans are capable of building. There are examples of this kind of complex self-assembly, though, that we can look to for inspiration to think about how this might work. What are the kind of physical rules that might be at play that allow this sort of near magical thing to happen? Here's an example that's appropriate for the Marine Biological Laboratory. This is a school of fish. Each one of these fish only has very local information about its neighbors. It really only senses its immediate neighbors. And yet the collection of fish are somehow acting together as a single whole, almost as a superorganism in this school as they move around. So there's something emergent that happened when we had individual fish knowing only about their neighbors that enabled this thing to act as a kind of a coherent whole. Here's another example that is appropriate for summer. We can think about a cold glass of water. And I'm going to show you this movie, and the first impression you might have is, gee, that's kind of boring. Like, I see that every day. I watch this happen every day. It's no less amazing. So what you're going to see here is in this picture, we can see the different states of matter that water exhibits in our daily lives. We've got the liquid state of matter, of water in this glass. There's also ice cubes, so that's the solid state, where the individual water molecules are kind of snapped together and register in this crystalline organization. And then there's the gaseous form, which you don't see in this yet. And that's a very low concentration. The molecules are all kind of unorganized and at low concentrations. So this is a time-lapse movie, and you all know what happens. Okay, it's a hot summer day, and what happens first is that the gas molecules somehow know to condense on the surface of this cold glass and actually start to collect and rain down. The solid form of water begins to melt, temperature's going up and it's melting. So just like in the example of the fish schooling, these individual molecules are changing their collective organization. They're changing the way in which they're interacting with one another to do something different, to organize and to self-assemble in particular ways. The gas molecules all condensing together into this liquid form, and the solid ice melting and so forth. So these are examples of what we would call phase transitions, where there's an emergent collective behavior that changes as a function of some external perturbation or external change. There's lots of examples of this kind of thing. I actually grew up in a field of physics called soft condensed matter. This is the physics of squishy materials. So I was a PhD student in the lab of Dave Waits, who's one of the pioneers of soft matter. And there's lots of examples of this kind of thing, these colloidal gels, particles that get together and form a solid gel, a soft solid. This is basically exactly what happens in Jell-O. I think we're all probably familiar with Jell-O from various wonderful cafeteria experiences. Foams, this is one of my favorite examples. Think about a foam is something like 95% gas, 5% liquid, and yet they come together and form what is actually a solid. It's a soft solid, but it's a solid. There's no chemical reaction there. It's just the way in which the liquid state and the gas state come together that actually makes a solid. And then there's things like this, these emulsions, liquid emulsions, or silly putty, also a great fun example that we play around in my house quite a bit with. So these are examples of emergent collective material states. And these are all examples that are non-living, but these are the kinds of materials that are most like cells and tissues. They're most like the kinds of materials that are living. This is something that was actually recognized by many of the early pioneering researchers at the Marine Biological Laboratory. Somebody who I'm quite fascinated by, there's a book about Ernest Everett Just by Kenneth Manning, which I would highly recommend to anybody interested in some of this history. But E.E. Just was a pioneering researcher who spent many summers here at the Marine Biological Laboratory about 100 years ago. And in this book, The Biology of the Cell Surface, Just asks the question of whether the protoplasm is an emulsion, a suspension of foam, or some combination of these. So he and others of his contemporaries, especially around the MBL, were asking these sorts of questions. Just was actually a bit skeptical about the application of ideas from physics and biology, and yet he clearly recognized that these kinds of questions about the kind of state, the material state, and what the cytoplasm or the protoplasm, as we call it, what that was really all about. Just received his PhD under Frank Lilly. Another of Frank Lilly's students was Victor Heilbrunn, who also spent many summers here at the Marine Biological Laboratory. He was a close friend of Just, and he was particularly interested in measurements of the material properties of cells, and did a lot of work with embryos and measuring their viscosity. And he viewed the measurements of the viscosity and the material state of a cell as really essential for understanding how life works. Roger Erlander Young was initially an assistant to E.E. Just at Howard University, and ultimately received her doctorate working with Heilbrunn at the University of Pennsylvania. She's the first African-American woman to receive a doctorate in zoology. This picture is taken just outside and the steps coming in, and you can go sit there right afterwards if you like. The picture was taken about 100 years ago, and not much has changed. There's some bushes here now. Roger Aylner Young has a sole author science paper about paramecium, and in that paper, she's also asking these kinds of questions about the material state of the cytoplasm, and she talks about this, whether it's a sol-gel transition, which is essentially a kind of gelation. So these sorts of ideas were everywhere in the air, in the discussions, particularly at the MBL about a century ago. One of the biggest proponents of this kind of thinking and using ideas from physics to understand biological systems was Jacques Lerb. He was a great admirer of the Swedish physical chemist Svante Arrhenius. Many of you probably know Arrhenius' law. And he was very interested in ideas from physics and trying to understand. And in fact, he views it as, has great hope that ultimately life, the sum of all life phenomena, can be unequivocally explained in physicochemical terms. So these were the kinds of things that were being discussed, again, a century ago. Jacques Lerb is meaningful to me because he, particularly because he was the founder of the physiology course, and my new historian friend Shane Jinson tells me that the first official real laboratory physiology lecture course was actually in 1899. I didn't know any of this history when I showed up to take the physiology course 107 years later. I was a physics student, becoming interested in biology, but trying to think about ways that I could contribute to understanding living systems. And this course was, transformative for me, and my career was an incredible experience. It was also important to me because it was here that I met my postdoctoral advisor, Tony Hyman. So I was a PhD student at the time and met my postdoctoral advisor that I then went on to work with. You can see the interesting lineages of scientific training because Tony did his postdoctoral fellowship with Tim Mitchison. Many of you know Tim. Tim and Ron's in this picture up here as well. You'll see a lot of people. I think Claire's in the picture, Dyke Mullins, and many others. But Tim and Ron were running the course at the time. I didn't know what I was going to work on, what I wanted to devote my career to at that time, but I knew that I was excited about biology. And if you go up to the library here, there's all kinds of wonderful things to look at, including this amazing plaque. Many of you have seen this it has a huge fishing lure in it with a massive hook. And next to it is this quote from Albert St. Georgi, who, as you know, was the discoverer of vitamin C and had done absolutely pioneering work in understanding muscle contraction. And he says that I would characterize my own research by telling you that when I settled in Woods Hole and took up fishing, I always used an enormous hook. I was convinced that I would catch nothing anyway. And I thought it would be much more exciting not to catch a big fish than not to catch a small one. I just love that. And I am a fisherman, and I can tell you I don't catch big fish very often. So Albert St. Georgi as well was affiliated with the physiology course, as by now you think just about everybody is. So I didn't know what I was going to work on. I knew I wanted to work on important problems. Sometimes important problems come in small packages, and I became interested in these structures, which are the organelles of a cell. These are basically as the name implies, they're the little organs of a cell. Think about the organs of our body that facilitate different types of functions that compartmentalize our body and enable the processes that are important for our bodies. That's basically what we think about in organelles in a cell. They're compartmentalizing the contents of cells. They're often speeding up reactions by concentrating reactants. Now, the textbook picture, if you Google organelles or whatever, or the things we're all taught in high school biology, is that organelles have membrane boundaries. They're like soap bubbles. They have phospholipid membranes. And that's certainly true for many organelles. But there's a broad class of intracellular bodies of these kinds of organelles that actually don't have membranes. And so these go by different names. We'll call them membraneless organelles, or increasingly we refer to these as condensates for reasons I'll describe. These things are really fascinating. And around the time that I got involved in all this, around the time I took the physiology course, not much was known about them except that they were, many examples in cells. These structures called nucleoli had been seen in a microscope for almost 2 centuries. There are examples like these signaling clusters, or I'll tell you more about P granules. They're dynamic, many tens of thousands or hundreds of thousands of molecules all in one little cluster, and they're highly dynamic, but there's no membrane. So the question was, why are they all together like that? What's holding them together? I became interested in this particular problem in C. elegans. So, the movie I showed you in the first slide was a C. elegans worm. This is the embryo developing into a worm. This is the embryo at the very first stage of its development. And something really fascinating happens where these green puncta that you see here, these green dots, which are the P granules, this collection of RNA and protein, all end up on one side of the embryo. So we asked really simple question: how do they all get to one side of the embryo? And in a series of experiments using advanced fluorescence microscopy, we were able to show that the process by which P granules end up on one side of this embryo is just like what happens with phase transitions of water. So if we imagine taking a box full of the gaseous form of water, so sort of high concentrations of water molecules in the gas phase, and if we were to cool down one end of the box, then what would happen is we would get condensation of these water droplets on this end, and it would basically pull the molecules over. This, hopefully you recognize, is just like the movie I showed you of this water glass where molecules are condensing on the surface. It turns out that this is not just a loose analogy. And as Dan mentioned, we came back to the physiology course in 2008. At this time, I was a postdoctoral fellow with Tony Hyman, and we came to teach in the physiology course. And together with some of the students in the course, we made the discovery that these P granules are actually liquid droplets of RNA and protein. They're condensed states of biomolecular matter where the biomolecules have all come together into liquid droplets. This is a movie that I made soon after the course of these P granules. You can see the white material here at a later stage in the worm, where they're all collected around these nuclei, and basically just squash this tissue. And what we could see is that these P granules, under these when they're squashed, they flow and drip and wet and coalesce with one another, just like the kinds of liquids we're used to, especially oil and water mixtures, things like that. We refer to this as liquid-liquid phase separation because essentially what it is liquid droplets of biomolecules, concentrated droplets of biomolecules that are coexisting with the surrounding liquid, which is a lower concentration phase. This is, in fact, exactly what's happening in salad dressing, oil-water mixtures more generally, where there are two liquid states that coexist with one another. The fancy word is immiscible. They're immiscible with one another. No matter how hard we shake this up, it's always going to separate out. Now, as you might have guessed, and with all the discussions going on at the MBL about the kinds of material that the cell or the cytoplasm or the protoplasm there were some really prescient thoughts along these lines at that time. This is from E.B. Wilson, 1898. He gave a lecture at the Marine Biological Laboratory. We're trying to pin down the exact date, but Shane thinks that it was probably a Friday evening lecture like this. And in that lecture, it was also later published in Science Magazine in 1899. He speculates that the living protoplasm is a mixture of liquids in the form of a fine emulsion consisting of continuous substance in which are suspended drops. So he has this thought that maybe there are, drops within the cytoplasm. I like this picture of E.B. Wilson because it shows that even in the late 1800s, photographers made scientists pose for ridiculous pictures. That's a tradition that continues. This is another picture. This is on the MBL history website. This is somewhere in the vicinity here. That's E.B. Wilson with a number of other pioneering early biologists, including T.H. Morgan. So I love this picture because, it's basically the same thing we do nowadays, right? We don't dress quite as nicely and we don't serve tea when we do it, but it's basically what's happening now still. So these liquid states of matter are really everywhere in nature. You're all familiar with liquids because they're everywhere. I won't list all the examples. Liquids are also very much of interest. I mean, there's still ongoing work to understand the physics of liquids, all these interesting things, and the physical laws that govern the Navier-Stokes equations and so forth that govern the motion and the dynamics of liquids are still actively studied. Liquids are also important for many industrial applications, particularly cosmetics, for example. So we thought, well, okay, these liquids are everywhere in the non-living world. Maybe they're also ubiquitous in cells, not just pea granules. So I came back to the MBL in the summer of 2009. Tim Mitchison often sets up a summer laboratory here. And I joined Tim that summer in his lab and started asking questions about these structures, these nucleoli. I already mentioned they've been seen for almost 2 centuries, but still were very mysterious. They're critically important for making the protein translational machinery, the ribosome, which is an absolutely essential structure in the cell. But it wasn't really clear how to think about how they do that or what nucleoli are as physical objects. So using a frog oocyte system, we were able to show that nucleoli also behave like condensed liquid states of RNA and protein. That's a movie of 2 nucleoli that had started to fuse, and I kind of pulled them apart. Now, my first PhD student at Princeton, Marina Ferrick, she went on to show that nucleoli, using the same frog oocyte system, that nucleoli are actually multiphase liquids. So this is a movie showing the green shows a subcompartment within the nucleolus, and the red is this outer layer. What this says in the end of the day is that there are basically a kind of a Matryoshka doll, a Russian doll nesting of liquids within liquids that facilitate this process of ribosome biogenesis. This looks a lot like the kinds of things that folks were studying in the Waits lab when I was there, these multiple emulsion systems of liquids within liquids within liquids. So this idea of what we call, as I said, liquid-liquid phase separation or liquid states of biomolecular matter is, we now understand over the last, 12 years, there's been from a trickle to a flood of papers on this topic. Examples include signaling clusters in the immune response, these structures called stress granules, various what we call condensates, this condensed form of biomolecular matter within the nucleus that are playing important roles in turning genes on and off, and literally dozens of other examples that we know of in the cell. There's a lot of popular press attention. If if you're a non-scientist, you might want to look at some of these articles. They're actually on our webpage. It's it's been fascinating. One of the things that's particularly exciting is this idea, this kind of soft matter physics idea, we can bring together with our understanding of the molecular details that make this all happen. And I want to tell you just about a picture that's important to keep in mind. It's this idea of interaction valency. It's a simple idea. So if we think about particles, maybe something like billiard balls, and they have the potential to interact with one another, but only when these two red patches see each other. So if I paint a little sticky patch on the surface of this ball, and then imagine throwing a bunch of these balls together, all that can happen is two of them can come together and form what we'd call a dimer. That's essentially a zero-dimensional object. It's not going to grow anymore. There's no more exposed sticky patches, and that's it. If I paint two sticky patches, I call that a valence of two, then I can start to get elongated structures like these filaments. And that's a one-dimensional object. It's like a line. And you can see that if I paint more and more patches, then I can start to get more extended structures and ultimately can get space-spanning three-dimensional networks of these particles. So that's an important idea in thinking about how molecules build these large-scale droplets. It turns out that one of the key ways in which we get high valence of molecules to build these structures is through polymers. Polymers, almost by definition, are multivalent interactors. They're chains of molecules. So if I think about them having sticky patches along them, then if I put them all into a beaker together, I can get an extended network of interactions that provides cohesivity to keep this droplet together. So that's an important idea in how this all works. One of the ways in which the molecules within cells do this is through proteins. Proteins are polymers. Even some biologists sometimes forget that. And the reason they forget that is because they're thinking about Emil Fischer's lock and key picture, whereas where we are imagining proteins as having a particular shape, and there's some complementarity of the shape for interactions. But it's ultimately a polymer, and the polymer can undergo conformational fluctuations, can undergo basically shape shifting, like you see in this movie, where it'll fluctuate around, not necessarily sit in one particular shape configuration. These kinds of proteins we refer to as intrinsically disordered proteins. Sometimes you'll see in the slides IDPs or IDRs. What we've learned, again, over the last decade or so is that these disordered protein regions are key for driving phase separation, for driving condensation into these liquid states in cells. Now, it's not just this conformational heterogeneity, this shape-shifting nature of many proteins. There are other parts of the protein that can contribute. Often proteins that are important in the cohesivity and holding these droplets together, they'll often have substrate binding domains, they call SBD here. So that would be something like binding to DNA or binding to some other type of molecule. And they often have oligomerization domains, the green region shown in the end there, the N-terminal domain. Those oligomerization domains are really important. And so just to give you a couple examples, this protein nucleophosmin has a pentamerization domain. So it sticks 5, very specifically 5 together. Whereas this stress granule protein G3BP has a dimerization domain. So it sticks 2 of them together. But we really didn't have many ways to study this, especially in living cells. There weren't the tools to interrogate this problem. And we were motivated by this kind of philosophy, encapsulated in a quote by Sidney Brenner: "Progress in science depends on new technologies, new discoveries, and new ideas," probably in that order. So what he means by that is often we scientists, we're just waiting around for new technologies or new techniques. And that, many examples of that: green fluorescent protein, pioneering work linked here to very closely to the MBL, super resolution microscopy, which has enabled us to see, with no longer fuzzy, foggy glasses, right? Seeing molecules within cells. Similarly, cryo-electron microscopy, and of course, the truly game-changing technology of CRISPR gene editing, which has far-reaching implications for humanity. So we were inspired by thinking about the technologies that allow us to do science, and my engineering colleagues and I at Princeton started to think about how we could control and interrogate phase separation within living cells. So this is some work that I'll in particular highlight. My former postdoc, Youngdae Shin, is now faculty at Seoul National University. The idea here is that we're using light to control biomolecular interactions and to control the oligomerization state of these often disordered proteins, these IDPs or IDRs. And so when we shine light on the system, we get these oligomers that then have the capacity to phase separate and form condensed droplets. These are some cells, and these are human cells in which we've expressed these constructs, and I'll shine light on the whole sample. And what happens is we get these droplets that form throughout the nucleus of the cell. Because this is light activated, we can turn the light off and on, just basically flicking the switch, and we can turn, cycle it, form the droplets, dissolve the droplets, ask about their reversibility. Are they even reversible? Do they disassemble when they're not oligomerized? And we can also do fun things like this. That's a smiley face, although I always think it looks vaguely evil. So we call it evil droplet man. But this is something that's still, we've done this work a few years ago now. It's still kind of mind-blowing to me. Essentially what we're doing is we're making it rain inside the cell, right? So it's been really powerful, these technologies, for allowing us to ask questions about how this all works, what's the molecular basis of the process, and how is it coupled to biological function. It's also been something that is really exciting for me because we're able to make maps in living cells for the first time that specify the conditions under which the system will form these droplets, indicated with the solid red circles, or the conditions under which they don't form the droplets, or the open red circles. That's called a phase diagram, and it's something that is common, ubiquitous, everywhere, and standard for non-living matter. But in biology, until we could do this, you really couldn't make phase diagrams for the matter inside living cells. What's also exciting about it is we can connect to theoretical frameworks from polymer physics. This is straight out of the standard polymer physics textbook, where you have a phase diagram that has that kind of upside down paraboloid shape, because it's ultimately reflecting the same kinds of physics. What's also, I think, striking though, and where biology can feed back into physics, is that these sequences are much more complex than any of the sequence in the proteins and the RNA and the DNA, much more complex, and the combinatorial complexity is much larger than anything in synthetic polymers, the kind of plastics that we know from consumer products. And so we're starting to be able to connect the sequence specificity with the detailed phase behavior and make these kinds of phase diagrams for different protein sequences. I won't go into the details of that, but just to give you a flavor of it. One of the things that's really interesting to us as well is the thought that maybe as great as these droplets in our cells are doing, I always say, don't freak out. You've got trillions of droplets in your cells right now. That's a good thing. But we wondered whether that could be tied to diseases. And in particular, we know that there are many protein aggregation diseases where you have proteins that all get together and form these kind of solid-like clumps in cells that mess up mess up cell function and kill cells and impair cognition and memory and so forth. In 2012, my postdoc Steph Weber and I wrote a review article in which we proposed the idea that these liquids, the condensed liquid forms that I've been telling you about, might serve as nucleators to promote solid forms that are irreversible and associated with disease, just like water droplets can freeze in winter, let's say, or if you put them in the freezer. In the summer of 2014, I came back with some students in my lab to the MBL. There was a summer institute that was established by Ron Vale, Jim Wilhelm, Mike Rosen. Mike Rosen circled there, and I'm in the back with the glasses. And other people you recognize, including Amy Gladfelter. And work that was directly started and based on the collaborations that summer led to a number of important papers that showed that these liquid droplets can, in fact, nucleate solid assemblies inside of them. This is an example in a protein We3 that Amy's lab had studied for many years, which is related to these polyglutamine proteins in Huntington's disease. There's another HNRPA1 and ALS, and Tony's lab had a paper that also came out on this protein FUS, which is also ALS related, where the liquids can form solids. There's actually a number of disease connections, really too many to even mention, but I'll give you some few glimpses. We know that the droplets that form, the condensates that form inside the nucleus of cells are forming in, on, and around the genome. The long polymers that encode genetic information. And they're turning genes on and off. They're controlling what genes are expressed, when they're expressed. And we know that that's at the heart of diseases like cancer, where oncogenes are turned on inappropriately and cells don't know that they should not continue growing and make tumors. So what's become apparent over the last few years is that many of these condensates are at the heart of cancer. Just to give you an example, without going into details, a paper came out last week in Nature that describes the way in which phase separation is associated with these genomic rearrangements, chromatin looping, and to drive cancer development. This is just one example, but it came out last week, so I thought I would show it. Here's another example, also timely given you're all wearing masks. I guess I was told I didn't have to wear one, but now I feel self-conscious about it suddenly. But the SARS-CoV-2, the virus we already talked about, this is some work from Amy Gladfelter, a friend and collaborator who spends summers here as well. This is a paper that shows that some of the components of the SARS-CoV-2 virus, the virus that causes COVID-19, undergo phase separation in cells, and that liquid-liquid phase separation process might be important for viral progression infection. There's many different viral infections that are now implicated, or where phase separation is implicated in the process. Oh, sorry, this is the movie I didn't play, but that's these SARS-CoV-2 component condensates in cells. Here's another example, respiratory syncytial virus that affects many children. The cells improperly fuse, and just, 2 days ago, Another paper came out in Nature. This is particularly fascinating, which suggests that there's a drug that they've identified which hardens these condensates, basically turn the liquid into a solid, and that hardening impairs the progression of respiratory syncytial virus. So all of these interesting disease connections. The biotech industry has taken note of the potential importance for therapeutics. And there's been a bunch of companies that have started to leverage these discoveries and think about new ways of treating devastating human diseases. I have a disclosure statement because I was founder of one of them. So taking it back to the beginning, I started out by telling you that living systems, cells, are technologies. They are technologies, though, that are vastly more complex and advanced than anything we human engineers can make. So when we think about maybe a picture like this, it's it's maybe misleading or imperfect in many ways. Living systems and the components within living systems are incredibly dynamic. I told you the numbers, millions of rotations per second, hundreds and hundreds of thousands of interactions per second. And so what we're starting to be able to do is think about the fundamental physical laws that govern the emergence of collective organization, the self-assembly of these molecules, in this case into these dynamic liquid states, which form compartments that are really at the heart of biology. This, I think, is exciting because it gives us hope that we're on track to one day meet Jacques Lerb's dream of determining the physicochemical rules that govern life. What is also particularly thrilling for me is to think about this kind of work in the context of the broad scientific tradition that goes back decades and centuries, much of it rooted here at the MBL. I told you about some of the work and thinking of early pioneers at the MBL, like Heilbrunn and Erlander Young and E.E. Just and E.B. Wilson, St. Georgie, and so on and so on. What I think is remarkable is that I've never met any of those individuals. They come from a generation or two before or more before me. And yet, I view them as colleagues in the same way that I hope one day future generations will see me. We're all part of this, what I think is a great quest to understand the physical chemical nature of life, which I think in the end is the question of what it means to be human. So I would like to thank you for your attention. I listed a lot of the folks that were involved in the work I showed you, but I won't read all the names. I want to thank some of the amazing current and former students, postdocs, staff, collaborators. I'd also like to thank funding, and you can read more about our work at our webpage, and I'd be happy to chat with you more about any of this. Thanks.
So people complain when we don't start on time, but they tend not to complain if we go a little bit over. So we're going to have a little bit of time for questions. I'm sure there are a few, hopefully in the room and online. So if you have a question in the room, just raise your hand. We're going to go back to the old-fashioned way here, and we'll walk around and pass you the microphone. And we might have some come in online as well.
Wonderful and clear presentation. Of course, lots of questions. So in this liquid phase, what allows or encourages, or whatever the right word is, the right molecules to find each other?
Yeah, it's a great, I mean, it's a great question. It's you know, the way we think about it is through the partitioning, the formal way, you know, partition coefficient, which is basically the probability that a molecule will go into a given condensate. I think that in some cases there's simple explanations, like if you have a condensate forming protein that has a substrate binding domain for some specific other molecule, then sure enough, when you add that other molecule to the system, it partitions strongly into the condensate. But there's other examples where it's less clear, and some really beautiful work on the physicochemical environment within the condensate by Tim Knott, where under some conditions DNA partitions in, under some conditions RNA partitions in, and we don't really have a good understanding of how to think about that. What is the environment that favors some types of molecules versus another? But certainly, if there are binding interactions, interactors of one type, then with a condensate forming protein, then they will get pulled in. There's also, I mean, I could go on about this, I won't, but there's size effects, and we've looked at some of that. So as some complex gets larger, it tends to be excluded from the droplets as it deforms more of the space. These are kind of entropic considerations.
Oh, hey.
Hey.
So condensates make me think about like weather, right? Water condensing, like the storm we had today. And this is often accompanied by temperature changes. I'm
Right.
wondering, are there examples where temperature will cause condensates to form or dissipate? And does this affect regulation? I'm thinking especially like cold-blooded animals, which experience very high shifts in temperature.
Yeah, this is a good question. And I think it's important because, in talking about phase transitions, one has to talk about water. And when you talk about water phase transitions, everybody thinks about temperature, right? So that's for sure. But, if you remember from your thermodynamics class, temperature is one variable, but concentration, for example, of impurities, we're familiar with that here in the Northeast because of salting the roads, right? To keep the ice down. That's freezing point depression. As you add more salt, you have to go to lower temperatures to get the ice to form. So in most of the examples that I'm talking about, temperature is probably not, a key variable that's causing things to assemble or disassemble. Rather, it's, for example, post-translational modifications to proteins, things like phosphorylation. I do think it's interesting, lots of, organisms that don't thermoregulate are subject to their environmental fluctuations in temperature. There's some interesting work there, and I think these Arctic hagfish, where, how do their eyes maintain translucence, and actually Amy had some interesting work on that with these different fungal variants that come from different climates and so forth. But I think most of the examples we talk about in the field are actually not temperature regulated. Yeah.
2 questions online. I think we'll take them in succession. And then if we have one more question in the audience here, I think we have time.
First one is, could you please elaborate on the ability of condensates to control gene expression?
Yeah, this is a great question, and it's something that is actively discussed and debated. And I think that the simplest picture, the kind of cartoon picture, which is probably at least partially right, is that what's happening is you have condensation of these proteins locally around particular genomic elements, and that concentrates other things that are involved in driving transcription. So in the case of, this is sort of a public lecture, so now we'll talk about some in the weeds here, but RNA polymerase II, right, the transcription machinery that turns DNA to RNA in general across the genome, those are probably being recruited together with transcription factors that have these disordered regions, things like FUS and BRD4 and mediator and so forth. In the case of the nucleolus, which is an example of transcription that's mediated by RNA polymerase I, we think of it in a similar way. You're concentrating the molecules, the enzymes, the factors that are key for making that RNA transcript. So, like I say, that's kind of a sort of the hand-wavy explanation, but that's the idea, is that you're concentrating, just like conventional organelles, the factors that are important for some set of reactions.
And the second one, you describe a sort of eutectic behavior of molecules in a fluid state. How effective is this process at excluding molecular types too, such as we see with eutectic behavior in metals and frozen solutions?
Yeah, so that's exciting. Somebody, bring up eutectic behavior. That's the kind of stuff we studied in looking at steel phase diagrams as a material science student. Yeah, I think that, honestly, we don't have that level of detail yet. These phase diagrams I'm showing are in a living cell, at least, it kind of is as good as it gets. We've got a good collection of phase diagrams for purified components. We worry sometimes about what that means, divorced of the intracellular context. It's much less crowded, and maybe you're missing things and so forth. But so I don't know. To my knowledge, no one's seen. If you're really asking about eutectic behavior, I don't think anyone has seen anything that is what you would really call eutectic in the traditional sense. But even if I would say we're not quite there yet, because the ability to do those sorts of measurements is that program is just getting going. Yeah.
Give me a workout. No, it's good.
So going back to the question about temperature, and then thinking about you personally, your own old work from when you were in the weights group, is the relevant temperature for phase condensation the thermal temperature, or is it the sort of like effective temperature caused by all the active processes going on inside the cytoplasm? Would those things affect the ability of these condensates to form or do transitions?
Yeah, this is one of the questions that is super interesting to me. People sometimes will say, phase separation is an equilibrium thermodynamic picture, and how does that jibe with the, fundamental out of equilibrium nature of life and so forth. But many of the examples that I'm talking about are really out of equilibrium. The P granule example, the first example, is clearly out of equilibrium because there's a gradient of stability of the droplets across the embryo. And the second example, this nucleolus example that we did here, is also out of equilibrium. It's driven by a flux of ribosomal RNA. So what Wallace is asking about is this idea that we think about temperature as a key variable. That's the whole idea with thermodynamics, right? It's called thermodynamics because we think about temperature and ultimately reflecting entropy and so forth. And that's what's driving the million rotations a second that I emphasized. But it's not clear that that's the dominant energy source and thing that's controlling emergent behavior in all the cases cells. So one can start to ask about whether there's something like an effective temperature. And that's the way we've been thinking about for the nucleolus. We were asking that kind of a question. You can measure, the way you do that, this is getting very into the weeds, but these fluctuation dissipation relations, and you look where they deviate, and that can allow you to assign an effective temperature to something. There's some examples of people trying to do things like that, but I think that that's still an area where there's a lot, I think there's a lot of room to get at the fundamental physics, which ultimately is about the physics at the heart of life, because it is out of equilibrium. So I think it's an important question. Yeah.
give Dr. Brangwynne one last round of applause.